Okay. All right. This is the last lecture of the summer school or the summer course, whatever. And I typically edit just talking about the future and whatever, but I decided to edit with this lecture. It's a little long. Hopefully there'll be a lot of information crammed in, but once again, I share all the slides with everyone in case you just want to have a copy. There might be some good references in here for future use. So this is one of my favorite talks to give, actually, because this is kind of what I think about all the time as far as my job is concerned. And my work is on functional MRI methods, which involves understanding contrast mechanisms, understanding applications, and everything in between. And what I'll argue here is that the field is pretty much driven. What drives fMRI? It's driven by the controversies and the mysteries and the challenges. That's sort of what makes it go forward. So let's see here. All right. So can everyone see that? So yeah, what moves the field of fMRI forward? So I would argue that the most important thing is the understanding that the field has that improvements in fMRI sensitivity, speed, precision, information content, repeatability, interpretability, all increase its utility. And that's sort of the goal, to make not only fMRI, but to increase the utility of fMRI to better understand the brain and for clinical applications. So at an individual level, how do individuals drive the field? Well, we're all driven by our own curiosity. That's more idyllic. We'd like to be completely driven by curiosity, but we're ambitious. Publishing papers gets us promotions. We get funding. At an institutional level, which we always have to keep in mind, it's mostly, how much funding can a person bring in and how and what themes they actually work on that fit in with the institution that's employing them. And with the research community, what drives the field? Well, certainly meetings, workshops, collaborations, standardization in tools and methods and platforms. That all drives the field forward because it gives us a way to communicate and share our data, our information, our code. And also platforms for developing novel tools and exploring the data also drive the field. Of course, people work with MATLAB, they work with other things. That's also extremely important for driving the field. But as far as the questions, in neuroscience, there's always a gap. We try to figure out what we're doing, what at least the PI is doing most of the time is saying, okay, what don't we understand? What kind of we can fill in with the tools that we have? And clinically, the gap is, how can we have some sort of insights that would help clinicians either give better treatment or, replace a technique that's more cost effective and with something that gives unique information that gives better treatment or better clinical diagnosis. So those are what drive the field as well, those questions, those needs. And at the funding source level, funders always think of, potential impact, whatever that means, and either short or long term and proposal quality and thematic category as well. And unfortunately, with the thematic category, over the years, 30 years of funding for fMRI, the methods themselves, even though they're fundamentally important, are never really funded in themselves. It's usually, if you write a grant, it's usually tied to an application or a biologic system or something like that. So something to keep in mind as you're writing grants is that people who do methods work, they kind of fall between the cracks. They have to figure out people to collaborate with to develop methods. So then there's always this natural tension between doing novel stuff that you think has high impact versus having it correct. And that's what causes a lot of these things that I'll talk about in the field. And I just sort of played with this idea. I was thinking, okay, so how do you illustrate how the field grows? And I'm thinking in terms of bubbles. And it started with fMRI, this sort of thing. that shows activation. But then it kind of, if you develop something like high field, it turns into a subfield in itself, and it branches out into new research avenues. And sometimes those research avenues branch into other things, like layer fMRI, and that becomes its own subfield. And the goal of all these subfields is sort of maybe to try to connect up with these two golden fields of clinical applications and neuroscience insights. And there's so far with fMRI, there have been some sort of neuroscience insights and maybe one or two clinical applications, but it's kind of sparse. We're sort of like a big field with not a lot of links to those things yet. So we're getting there. So I showed this before, the 4 fMRI pillars. These are the categories that I'll just talk about briefly in terms of talking about the impact of fMRI. So the categories are, starting with, and also if you do work that's as much as possible at the interface of what the best that the categories offer, the papers are usually the highest quality and the highest impact. So we rely on pulse sequences and hardware. Vendors supply those, but we try to develop it. We also care about understanding what the signal means. This is the pink bubble. We do a lot of development in paradigm design and processing methodology. And that all ties into all these other techniques. You develop your paradigms and your methodology based on what you have and how well you can interpret your signal in terms of the hardware and the interpretation. And of course, you would like these to feed into clinical applications and basic research, basic insights about the brain. So you don't have to, I'm not going to talk about this whole timeline at all. I'm just showing it as a sort of display. It's my own pet project to sort of add to this. But you can look at in terms of pulse sequences and hardware and interpretation, contrast mechanisms and physiology, as well as paradigms and processing, those 3 things. I've sort of left out applications because that's huge. All the types of things that have been developed as a function of the year. So this is from 1991, when fMRI began, and all the way to now. And it's not like it's falling off in enough things. So, and you can see there's this steady progress of these interesting things that might in themselves represent a little bubble in that diagram that might start a subfield. So everything from processing methods that have evolved, standardizations, things like that, that have all evolved over time. But to simplify this plot, the development of fMRI sort of followed this thing, this simplified time series, the time course here. And 1991 to 1996, everyone was just amazed. It was like, wow, we have a signal here. That shows what's going on in the brain, non-invasively. This is amazing. So everyone was really happy with that. They were just mapping blobs, like doing cartography, saying, oh, tap your fingers, there's a motor cortex. Amazing. The neuroscientists were sort of thinking, this isn't that amazing, but it's great that you could do it in humans non-invasively. And then resting state was discovered in about 1995. That started a whole subfield. Standardized fMRI processing platforms, which really pushed the field forward, started. And then about 2001, sort of catalyzed by Logothetis's work and other people's work, people started to believe that fMRI actually reflects neural activity. They were doing simultaneous measures with other modalities, with electrophysiology, showing, oh, neural activity, fMRI. So people started to believe it. This is also when large databases began. People started, instead of doing blobology, people were doing network mapping. And then the fields, once they got tired of doing all this activation studies, they didn't get really tired, but I mean, they started to finally move into resting state connectivity. Resting state was pretty dormant from 1995 to about 2005. Nobody really paid much attention to it. And then now that we have sort of modern challenges of how to do integration of multimodal information. We have more of a accelerated dive into spatial and temporal richness and the granularity of information. The MRI technology continues to improve. And just to emphasize here, it's not improving because fMRI necessarily needs it. It's improving because the vendors are seeing value in improving the technology that fMRI then uses. The vendors, as I mentioned before, don't really care about fMRI. And there has been a jump in the overall sophistication of the analysis. So that's where the field is at. But there's still a lot of questions. And I'll get into these later in the talk. Do we have biomarkers? Biomarkers is this sort of buzzword. Like, it would be great to put a person in a scanner and say, you have schizophrenia, and this is how we're going to treat you. Or you have, any other disorder or shade of disorder. We can't do that yet. And the big problem isn't because fMRI is not sensitive enough. And I think this is a point I really want to bring home. I'll emphasize a couple times. It's not because fMRI is not sensitive enough. It's because, first of all, we haven't quite figured out what those biomarkers might look like. And the other thing is that there's so much variation among brains, among individuals, even with these disorders that manifest differently in the brain, that we haven't yet got our heads around those differences. So that's partially why the field is going into looking at individual differences, just to try to get a better handle of the variation among individuals. So, people are similar in terms of just very basic things. Motor cortex, is in the same place. But when you talk about a mood disorder or other things like that, it gets more subtle, a lot more subtle. Big data analysis, we need to have better curated data. Individuals, as I mentioned, that's increasingly more important and interesting. Clinical utility, not really yet. I mean, pre-surgical mapping is good, but it's not really used clinically yet. Layer fMRI is promising, but it's not quite there yet. A lot of challenges. Dense sampling, the idea that you take one subject and average the heck out of the data so that you can really see the subtleties of the networks. And that's becoming more important as individual subjects and their differences become more important. We can look at individual differences without dense sampling, but it sure does help to have that. Multimodal imaging is still becoming increasingly necessary for either understanding the noise and variation and regressing it out, or just better interpreting what the signal means. Of course, machine learning, AI is just getting started. There's some manifestation of that now in foundation models. You've heard that buzzword. I'm still trying to understand exactly what that means, but that's becoming huge, and it could take over everything, and which might be a good thing. Who knows? All right, so some significant advances over the years. Let me just I screwed up with that slide. I'm not going to go into detail with all of these, but there's been advances in pulse sequences and hardware, paradigms in processing. These are sort of like the landmark advances. And like I said, these are sort of like things I'll just briefly mention. You can refer to the slides later for references. Interpretation of the signal and applications. And then, without going into too much detail, we've generally gotten higher resolution, we've gotten faster in pulse sequences. Paradigms, we've been able to develop ways of observing the brain, manipulating it in time such that we can pull out ever more subtle aspects of neural activity, like adaptation, for instance, other things like that. In interpreting the signal, we understand that there's certain types of neuroactivity that better reflects, that is reflected in bold contrasts, like gamma frequencies and things like that. And of course, applications, the idea that we can actually make whole brain maps where no other technique can, other than PET, which is a little bit slower and more invasive, that really has helped a lot in terms of a lot of things in terms of applications and pre-surgical mapping. But of course, there's a lot of other applications that are sort of in the pipeline: real-time feedback, neural feedback, longitudinal studies, intervention measures. Those are all look very promising as well. So the common criticisms of fMRI are that the signal's slow in course, it's variable over time and space, not fully understood, and not directly informative of neural mechanisms. And mechanisms I mean by the scale right below what we can measure with fMRI might be informative as to what's actually going on. What does a computation look like? When you move your fingers, you say, oh, that part of the brain is active. It tells the motor neurons, the motor cortex, or the fingers to move. But we say that, but there's a whole lot of stuff in there that we have no clue as to what's going on in terms of how neural activity actually translates to moving the fingers, and how our decision to do that translates to the motor cortex to do that. So fMRI can't get anywhere close to that. So mapping doesn't give insights into brain mechanisms and principles. The images suffer from signal dropout, sensitive to motion, all the standard things that we haven't fully solved yet. And but it is sensitive. It's extremely sensitive, and it's robust. It's not invasive. It's replicable if done correctly, and frankly, it's the best thing we have. So that's why we're using it. If there's a better thing that comes along, fMRI will probably go by the wayside, possibly. But it's the best thing we have. It's been the best thing most of us have had for the last 30-some years. So most fMRI research has been towards understanding functional brain organization. And the desire and the thought is that the signal means something. It contains information. It's sensitive, precise, and repeatable, and the spatial and temporal scales are relevant and useful. Even some people might say, well, since you can't look at actual neural firing, then it's useless. A lot of people have very reductionistic views of the brain, whereas I would argue that most of our behavior is on a time scale that's perfectly suited for fMRI. It's everything from, if you're looking at the time scale here, it's everything from perception. We perceive things on the order of maybe 200 milliseconds or so, and we can measure a signal that's generated from a 200 millisecond stimulus, all the way to the really long-term stuff, learning and plasticity over time, over years. We can study that with fMRI. And spatial scale, certainly fMRI covers a lot of things. This is the temporal scales that fMRI covers. This is the spatial scales, and you saw this already. But essentially, we can get all the way down to sort of the what's called the mesoscale. It's sort of the circuit level across layers or columns, which actually might be the essential building block of cognition and other parts of the brain. Things might be more homogeneous below that. So less information about how the brain is organized might not be below that scale. So we might be exactly in the sweet spot spatially, to be honest. So why do we want to improve it? Well, there's more information in the signal. If anyone looked at an fMRI signal, you see all kinds of stuff. You see the activation signal varying over time. You see the post-undershoot. You see transients. And you see a lot of noise as well, which we're still trying to model and get rid of. So that's kind of the drive for a lot of us, to better understand the neural correlates of the interesting transients, and also to try to better model the noise so that we can get rid of it. That pretty much sums that up. So once again, fMRI doesn't suffer from a lack of sensitivity. You can actually get very amazing activation within minutes of most brain activation. And we just haven't figured out to fully wrangle the data from the backdrop of variability. This is in time, space, and also across subjects. So a lot of variability. Haven't quite figured it out, the source of all of that enough to model it. So over the years, I did a rough search, and it seems like there's about over 80,000 functional MRI papers that are published. So what's been the impact so far? One big impact has been dynamic connectivity. has sort of catalyzed sort of a new way of thinking and a new way of doing research in multimodal human brain research. And this might seem so obvious to everyone. It's almost like a blind spot. But before fMRI came, we weren't thinking in terms of networks. I mean, if you do electrophysiology, you study one neuron at a time, you don't know what the rest of the brain is doing. When you do PET scanning, you have sort of blobs, but you can't really see a dynamic network. So fMRI really captured that. So it established the brain network perspective and its relationships to all kinds of things: tasks, behavior, states, traits, spontaneous engagements, pathology. But once again, doesn't yet provide sufficient. It doesn't provide mechanisms or insights for clinical utility yet. So fMRI has also allowed us to identify changes with learning, adaptation, plasticity, and so on, to help better understand these processes, such as attention, language, sensory integration, emotion, and cognition in general. So we're able to look at these changes because it's so non-invasive. You're able to look at these changes over time, which is not a trivial thing. So, and then it's also catalyzed the rethinking of individual differences and similarities and the categorization of pathology. So one thing we're finding, there's two initiatives. I mean, there's RDoC that started about 15 years ago, and there's been the DSM sort of categorization of psychiatric disorders that's been developed over the years. And what people are trying to do is have DSM sort of more driven by the data in the brain. That's driving a lot of grants. That's driving this RDoC initiative. It hasn't quite been useful, but it has found that categorizations should be more finely delineated than we have thought. So the data are emerging that we probably need to rewrite our DSM manuals. So and also it might sound trivial, but fMRI sort of catalyzed, I think. It still was going on, but fMRI has catalyzed research in neurovascular coupling, brain energetics, as well as CSF and vascular physiology. So that was going on, but the fact that fMRI is so sensitive to these things and it's so important to interpreting the fMRI signal that it sort of catalyzed that research as well. Okay, so I'm going to show this plot again, but add the controversies. So the controversies are kind of fun and sort of interesting, and sometimes they're frustrating, but they also drive the field. Sometimes something comes up as a controversy that somebody makes their name on saying, hey, this is the problem. And they become famous, and then the field has to rally around that problem to try to solve it or figure it out or move beyond it. And so very early in the field, it was the question of, what are we looking at here? Is it just all veins? And how does that reflect neural activity? Is there an issue? And then there's some aspects of the signal, the initial dip, the post-undershoot, there's white matter activation, and I'll get into some of these, the dead salmon controversy of false positives with multiple comparisons, and so on. That sort of caused the field to sort of say, wait a second, we got to solve this, and it sort of pushes things along. Okay, and I categorize the controversies in three categories. One is practices that might be incorrect or misleading, or discarding information that might be useful. Also, shortcomings or curiosities that are just amplified by non-experts in the media. And a lot of times, you have some science article or whatever that says, 20,000 fMRI papers are wrong. That's causes most people in the field to sort of shudder, to think, okay, that's not right. And now the whole field, including funders, hear this. We got to do some damage control here. And also non-reproducible findings. Sometimes people have tried to look for activation-induced changes in diffusion, neural currents, et cetera. In white matter, pre-undershoot related stuff, small n group comparisons. And this has been very prevalent, and it's a good thing that we got rid of that. So yeah, all of these are considered controversies. And I just want to pause with the small n group comparisons. You might get findings, but you amplify the effect size when you do that. I don't know if there's been a talk about that, but I was actually just talking to Danny Pine. And this was kind of a shocking thing the other day, where if you know who Danny Pine is, he's probably one of the more prominent fMRI neuroscientists, clinicians in the NIMH. And he had some hugely high impact papers between 2006 and 2009 that were all relatively small n papers. And so I asked him when I was interviewing him on this podcast, I said, oh, well, what do you think of those papers now that there's been, after these sort of things have come out to say that you need a larger n? And he's like, oh yeah, all those papers are wrong. and I'm like, whoa, it's and he didn't hedge, and they're not wrong. And so it's a little bit more nuanced than that. They're not necessarily wrong in terms of what they imply, but wrong in terms of the estimation of the effect size. That's never estimated properly with a small n, no matter what. Can't get around it. So, but are they useful? They still might be useful to catalyze future research. But on the flip side, and I'll talk about this too, there was a recent paper by Merrick that was published that said, oh, wait a second, you actually need 2,000 subjects to say anything. And while that was a correct paper, that was actually a really good paper. It sort of set things up a little bit as a straw man, because there's a lot of reasons that it sort of the data sort of shot itself in the foot in some sense with pooling together different types of pathologies in terms of psychosis, looking at just resting state, not doing multivariate comparisons, not having issues in registration of the brain. So the real answer is somewhere between 2,000 and 200, but it might vary depending on how good you are in getting rid of all the variations. So that's an important aspect. But either way, all right. So I'm just going to briefly talk about these sort of things. So in 2012, there was this thing that actually won the Ig Nobel Prize, and it was sort of silly. It still is kind of silly. It's people decided to put a dead salmon in the scanner and see if they get brain activation with visual stimulation, and they found a few voxels that were activated. And then the media just gobbled this up, and it sort of was made fun of fMRI. It's like this silly thing, but in fact, it's a real issue that everyone who knows fMRI just knows that if you look at enough voxels. If you have thousands of voxels that you're looking at, by chance, you'll have some that show significant activation. And so you have to do what's called a multiple comparisons normalization. And every statistician knows this, but still it got attention. This was a little bit more significant. This actually changed the field a lot. It caused people to be aware that you can't, the number of papers on this, but the idea is that you can't do circular analysis. You can't select the data and then look at the effect from the data that you've selected using the same tests. That boosts your correlations. So that's why everyone needs an independent data set doing their analysis, and they do the selection of the data either for an ROI or whatever with a different set of data, or they do a different training set for something else. So that was important. There was the clustering conundrum, and all these have names. This one, and it's a little bit nuanced, but I won't go into exactly all the details of it. But essentially, there was some controversy as to how statistically you clustered your data, and some cluster sizes were generally a little bit overestimated, and due to autocorrelation in the data. that didn't follow just an assumed Gaussian shape. I mean, the data is not perfectly Gaussian. So, and this is the one that there was a blog post written by Tom Nichols saying, 20,000 papers are wrong. And then the media picked that up and it's like, oh, 20,000 papers are wrong. I'm like, well, okay. So 20,000 papers might have a reporting of a cluster size that's a little bit bigger than what you expect. And then I challenged anyone. I still have yet to see any paper. Did that ever change the conclusions of the paper? And the answer is no. Not one paper was affected in terms of the message that it conveyed or the conclusion. So if people did this correction, would the results be different? Would the conclusions of the paper be different? And the answer is no. So that's an example how a controversy, which is important, and which was all the platforms actually did the correction to address this, wasn't that important. All right, so there's other things, and this is one that we kind of got embroiled into a little bit, was there's a study, electrophysiology study, in which Siriton and Das did this, basically said, we're seeing that when these rodents, I believe, they're doing electrophysiology studies and hemodynamic studies, and they were getting sort of habituated to the stimulus. And then if you didn't give a stimulus and they were still expecting it, they claimed they saw a hemodynamic response, but no electrophysiologic response. And they said, look, we found something, that we get bold signal without any electrical activity. And there was some back and forth. And I think ultimately the conclusion was that there still is some electrical activity that drives the hemodynamic response. It's not like just driven by something else. There's not some other magical component that drives the hemodynamic response. So, and this is also a recent paper that kind of caused people pause, and it's an important paper. I mean, this paper is also a correct paper that, in some sense, it's called the NARPS paper, and I'm not sure what the acronym is, but basically, it was a study where they gave a data set to over 90 groups, or 75 groups, or 72 groups, or whatever, and they all did their independent analysis on this data set. And I actually was a reviewer of this paper, and I warned them. I said, this is, you're probably couching this in a way that will get popular press to jump all over this, because their conclusions were that 70 teams were analyzing the data, basically, and it was some complex tests, some basic tests, some complex tests, like a gambling task, and they're testing, predefined hypotheses. So with different regressors, they put in hypotheses, and their conclusions were that the results were all over the place, all different results. And then people who didn't actually read the paper carefully would think, what does this mean for fMRI? There's results all over the place. And the point that wasn't brought out in this paper, that was brought out very nicely by Paul Taylor and others who were writing to address this, was that if you lower the threshold a little bit on this, just a little bit on this data, or you have unthresholded statistical maps of the data, and then look at the correlation across all the studies with that, they show a super high correlation across all the studies. So the statistical maps themselves were highly correlated. It's just that based on the maps, some shows a different threshold. And so even if you did motor cortex studies with multiple subjects, and if you raise your threshold high enough, every subject will have a different motor cortex location. But if you lower it just a little bit, it all clumps in the same area. So that's sort of what this paper brought out. This blue line is just simply some of the groups that were kind of clueless who actually had the regressors reversed. So I don't even know what the groups are, but they just made a blatant error of regression sign. So anyway, And so this is the Merrick paper, as I was referring to. That was a controversy. It wasn't a controversy in itself. Everyone read this paper and were like, "Oh God, that's not good." But it's a good paper, and basically saying that the effect size is so small that the statistical significance only reaches some sort of reasonable level after like about 2,000 subjects are averaged into this data set of comparing like psychosis with normals, looking at resting state. But then as people looked into this, one, it's not a problem of fMRI sensitivity. It's more a problem of the variability across subjects, variability of resting state, variability of normalizing each brain, variability of the psychosis. So, and there are several papers that talk about various errors one can make in their analysis. So it was a nice paper that was sort of a line drawn in the sand saying this is what you need to consider when you're doing your studies. And we still haven't solved this. It would be nice to someday see the study come out saying, oh, if we did this, and this correction, you actually get the same significance after about 200 subjects, which I think will happen, but we're not there yet. So, okay, some of the mysteries. All right, those are the controversies. So I started with the controversies. I'm not going to go over all the mysteries because the mysteries and challenges even grow more. But so what are the mysteries? Why does a bold response even happen? And I thought this wasn't a mystery for a long time, but then I realized it actually still is a mystery. There's still people writing about basically saying that bold, and I talked about this in one of my first talks, that it's not really driven to deliver more oxygen to active tissue. It's maybe to remove waste or keep the pH the same. Because if you vary your pH in your blood around and you keep other waste products there, that damages tissue, much more so than a lack of oxygen when it's active. So what are the mechanisms behind bold transients, nonlinearities, and inhomogeneities? We see all these nonlinearities and inhomogeneities in the data, and we still don't fully understand what the neural source of those are. So that's what kind of drives me every day. I sort of think of these things. I'm thinking, oh, how do we extract this information? And there's something like, for instance, inhibition versus excitation, which every neuroscientist knows about. The brain is mostly inhibitory. It's most of neural activity is to inhibit the brain from going out of control. Otherwise, you would have seizures all the time. So, but we can't differentiate between inhibition and excitation. So when you move your fingers, you have to have an excitatory signal, but then you have tons of inhibitory signal that keeps everything sort of stable. And I think those are differentiable. And it's a matter of, in fMRI, it's a matter of just understanding what the signatures of those are. So what are the sources of the spontaneous fluctuations in connectivity? And what is the global signal? That's another sort of controversy that I didn't talk about. People regress it out or not. People think it contains information on vigilance and other things, or they don't. And so we still don't really understand all the types of spontaneous fluctuations and all the and those changes. So what's the purpose of resting state fMRI? So the purpose, I mean here in quotes, meaning the evolutionary purpose. Is it just an epiphenomena? The brain's just neurons just firing spontaneously, or does it serve a role that's important to like maintain homeostasis in some sense? And we don't know that. All we know is that it really works well, and we're using it. So, and it gives us information, but we don't understand why it's there. So what are we missing in activation? So we typically do a lot of different regressors to pull out with block design or event related, but there's a lot we're not modeling that's there. That's I'll demonstrate just briefly what I mean by that. Why is the parcellation size what it is? Are there finer parcellations? Can the brain be parcellated all the way down to columns or layers? And what's a meaningful parcellation? Do parcellations vary across individuals in time? That's less of a mystery. they do, but just what drives that? How much is noise? How much is interesting information? And what's the best way to compare brains across and within groups and across individuals at different resolutions? Once again, that's maybe more of a challenge than a mystery, but and how relevant, and this is kind of a mystery in some sense, how relevant is the spatial and temporal scale of fMRI for deriving principles of cognition? You know, we all want to just understand the brain, whatever that means. We want to understand the brain in the sense that we want to know, not only describe what's happening when it does things, we want to understand at a mechanistic level how it actually works. And, you know, we can do that with muscles, we can do that with the heart, we can do that, you know, we understand the body pretty well. The brain, we have no clue. And there's still principles to be found. Just like, you know, in the 1800s, Darwin discovered principles of natural selection. Before then, everyone was just like, oh, wow, there's lots of variation. We can describe it really well. We have no idea. Wow. And Darwin sort of laid out this principle that sort of made everything make sense. We don't have that in the brain. We just don't. And can fMRI really help us? So here's an example of, I'm just going to go through a couple of these really quickly. I know I want to make sure I get done as close to 2 as possible. This is the paper. I'm just pointing to it. It's basically extensive modeling, but here, very prominent people in the field, very respected people, argue that CBF changes versus CMRO2 slopes, basically arguing for the idea that it's to maintain homeostasis of pH and partial pressure of CO2 and O2. So not necessarily to deliver oxygen to the furthest neuron from the capillary, but more to extract the CO2 and extract waste. Non-linearities are all over the place. I gave a whole talk on this. I'm not going to go into any detail, but they exist in terms of time, in terms of stimulus intensity, spatially, they vary. And they vary with duty cycle as well. So, and there's all kinds of transients that we're still getting our head around. You know, the pre-undershoot, the post-undershoot. Pre-undershoot is sort of variable. Post-undershoot varies with stimulus. It varies with contrast weighting. It varies with a lot of things. We still don't understand that. And the question is, why do we really want to understand that? It sort of seems like slightly esoteric. Why do you pursue these little things? Well, those could end up being interesting features that might give insight into pathology. You know, some people may not have as much of a post-undershoot than others, and then we'll know why. And so it might be all these different subtle aspects that we're trying to study are potential levers into adding insight about the brain that might be useful clinically or if we're understanding the brain. Inhibition versus excitation. So there's several studies that are just beginning that are using optogenetics and also things like GABA, you know, just injecting GABA, which is a inhibitory neurotransmitter. Inject more of that, you get less of a bold signal and you get a little bit more of a pronounced transient at the beginning. If you do optogenetics and selectively stimulate excitatory versus inhibitory neurons, you get hugely different fMRI signal changes that show this is with inhibition, you have a brief transient, a large post-undershoot, and here you just have, you know, your normal activation with activation. So this is just beginning, and we're still getting a handle on this sort of thing. So as far as the types of fluctuations, so what contributes to spontaneous fluctuations? Well, we know that there's a neural component, there's a respiration or cardiac component, and there's thermal noise and motion and scatter instability. So we have those 3 components right there. But with the somewhere between here and here, it's still kind of a mystery. And this is some work by Javier that sort of reflects some of the work by Laura Lewis, showing that there's these low frequency components that are at the base of the brain that might be a little bit of CSF being driven by neural activity. When you're drowsy, that might drive the lymphatic system. And that might have a correlation with negative correlation with everywhere else in the brain. So that's something. That's a much lower frequency than standard bold resting state fluctuations. This is a beautiful paper, one of my favorite papers by Logothetis, where he was measuring the hippocampus during sleep. And he found sort of the same thing as we found here. I mean, I wouldn't say it is the same and that what we're seeing is this, but very similar. He looked at hippocampal ripples that spontaneously happened when a subject was sleeping and used that as a regressor and found whenever a ripple occurred, the base of the brain, all the subcortical structures showed negative correlation. The cortex showed positive correlation. So that's, yeah, you could come up with a hypothesis for what that is, but nobody really knows. Then Shelley Keelos has sort of studied extensively quasi-periodic fluctuations where they're low frequency and they don't organize necessarily in the way that parcels do in the brain, and they might reflect another process, other processes related to their vigilance, somewhat related to the global signal, and it's still a source of, and it organizes across larger spatial gradients in the brain. So that's sort of a mystery still. And Josh Vaskowitz actually gave a talk on this, and this is something that I think is kind of lost in the field potentially, is that resting state fluctuations. We always characterize it, you know, the signal itself oscillates. If you do a power spectrum of the signal, it looks like it's 0.2 hertz. And when people think of 0.2 hertz, they think, oh, it's an oscillation. And that's just a bunch of oscillators that go at 0.2 hertz. And what Josh has shown, and you know, others before him, including you know, people he's collaborated with, Rick Betzel, and so on, that that if you do edge analysis, you basically take the product of every single, if you divide the brain into parcels, you take the product of every single signal on a time point by time point basis, which is like having a sliding window that's a width of 1. You find that the correlations look very different at different moments in time. And this is just getting to the punchline here, since I'm running short in time. Essentially, if you take the correlation of the data, and for instance, you look at the top, you look at the functional connectivity, the mean of everything, versus if you just take the top 3% of the data and look at the function and look at the average of that, you get a very similar spatial map. And also you can count the number of events and have a similar spatial map as well. So basically the argument is that resting state connectivity is not this sort of, you know, slow oscillations that sort of build over time in the brain. It's like these punctate events that occur that consist of very small fraction of the time series that might be not all of it, but the biggest driver of functional connectivity in resting state. So that's something new that's still, you know, it's there, it's out there, the field knows it, but we still haven't quite fully tied together everything in terms of understanding it. So the purpose of resting state fMRI, I just put this slide out here because I find it interesting that if you look at, for instance, and this is an example that sort of suggests different purposes in some sense, that this is some nice work by Newbold at the WashU group, basically if they put subjects' arms in casts, immobilize them, within hours, the resting state connectivity between left and right motor cortex drops. And if they take off the cast 2 weeks later, within hours, it goes back up to normal again. And during this time, every once in a while, there's this pulse that's basically a probe pulse that tries to see if there's a connectivity. My argument for that, I think, is one that says, well, resting state might actually reflect more of what has happened just in the past few hours. I mean, it's accumulation over time, but it might be the networks of the brain sort of, you know, optimizing themselves after they've been active in some sense to sort of find the lowest dimensional sort of path to fire so that they're able to fire more readily later. And this is an example of where we looked at the resting state connectivity between left and right motor cortex, and we find this is just very preliminary data that we collected. And when you look at it, you're like, oh, okay, it looks kind of like left and right motor cortex as you move a seed along is correlated. But if you look at the magnitude of the correlation, it varies a lot, and it seems to drop dramatically at the hand area. It's still high enough to show a correlation, but it's much lower than everywhere else in the motor cortex. And at least my very rough hypothesis for why this happens is because in the motor cortex, almost everything is symmetric in terms of how you move it. It's like when you talk, your left and right mouth motor cortex is active simultaneously. When you walk, your legs are, all active simultaneously. But hands don't really work simultaneously. They work sometimes together, but they're the least correlated. And so this is kind of cool then, if that's the case. It seems like a very sensitive measure to how much each part of the brain does work synchronously in reality. All right, so great paper. I think I already mentioned this, but by Pizzullo, 2021, kind of an open-ended sort of philosophical paper, but really nice. Basically, as I mentioned before, it posits that it's all about why the resting state occurs, why resting state connectivity occurs. And it basically says, might reflect, slow transitions between generic priors. Spontaneous neural patterns may reflect slow transitions between generic priors tuned for low-dimensional general-purpose behavioral patterns. And the dynamics across sequences of brain states that reflect exploration of the brain's dynamic repertoire to remain flexible. So basically, after the neurons are active together, they sort of start spontaneously firing to find a better connectivity path that's more efficient so that they can activate better later. So that's a very hand-wavy way of saying that, but it's sort of a network optimization. That's not really understanding, but it's sort of a hypothesis. So what are we missing in activation? Very quickly, here's an example of just modeling the transients. So this is really nice work by Camille Uladog. Very simple paper and kind of an obvious paper, but I just love it. Whenever you show hemifield activation, it's a classic experiment where you show a person one hemifield and then the next hemifield, and you say, oh, right hemifield activation, you get left visual cortex. Left hemifield, you get right visual cortex. And you put in a regressor that shows when the stimulus is on. Makes sense. But what they show here is that if you actually use the regressor that had the stimulus on, you get your left and right visual cortex. But if you put in a regressor that just represents the transient signal at the very beginning and at the very end, you get a bilateral activation that's very structured. So this is real activation that just missed completely because they chose, most people choose to put in a simple regressor. So this is something to think about when you're designing experiments and how to design your regressors so that you don't miss anything. There is a problem, on the other hand, of overfitting, but that's another issue. And I always show this because it's one of my favorite papers as well with Javier Gonzalez Castillo. Basically, he averaged the heck out of the data, had subjects in there for 9 hours, and basically showed that all of the cortex, with a very simple task. You look at a stimulus, you try to see if it's a letter or a number, you click a button. Very simple task. It's sort of a block design. The standard areas are active, but after you average for 9 hours and have an open-ended sort of just looking for anything that's time blocked, you find crazy looking signals, mostly negatively correlated, but all over the cortex and organized in such a way that it's not really noise. So the whole cortex does something when it's presented with a task, whether it could be transient, it could be negatively correlated, but that's just showing that there's a need to either average more or then have regressors that are more open-ended. I won't go into parcellations. Basically, there's evidence that there's many different parcellation schemes, and really nice study here. As the parcellations get smaller and smaller, the homogeneity within the parcellation, as expected, gets higher, meaning that there's still heterogeneity that you're averaging out with your parcels. So the solution is not to look at, people parcellate the brain to do resting state connectivity analysis, but that if you have enough signal to noise, it's always better to go finer resolution, all the way down to probably columns. Here's some work when we were looking at resting state. We did ICA in an iterative fashion, such that we first selected, the motor area, and then from the motor area, we selected ever finer and finer ICA components, and we were able to get down to ICA components that were represented each digit of a finger. So the brain is obviously organized in this fine scale. It helps to have parcellations that match that. And each individual parcellation is unique. So when you try to just put things into a template, when one tries to put things into a template to average, just know that you're averaging out all the individual information and losing a lot. Really nice papers on that. One way of possibly correcting for that, this is by Evan Gordon, basically showing that the parcellation pattern follows myelination, the myelination pattern. So you can make a myelination map with MRI, and you can potentially normalize based on the myelination map. So, okay, I have 1 minute for the challenges. So I'm not going to really go into this, but there's a lot of challenges. I'm not going to go into all of these, but the idea here is that we have to understand the noise. And here's just basically illustrating the problem, is that this is our signal that we're interested in. This is the correlation signal. This is the nature of the physiologic noise. It's highly correlated in the frequency spectrum. So we can't just simply differentiate it. We can't take a power spectrum and say, oh, let's just filter it out. There's no way. It overlaps so much. We have methods of doing that are different. We have multi-echo, which Dan Handwerker and others have talked about. That produces nice maps. And I don't know why I did this. There's many components in the fMRI signal that affect its magnitude. And we need to figure out, this is a challenge to how to normalize for these things that affect the magnitude of the HRF. Things like blood volume H voxel, things like the neurovascular response function, and so on. And I had a method a long time ago, basically a dividing from a CO2 stress or global stress versus not, to get a better normalization technique. And this becomes more relevant when you're doing layer fMRI, since you're highly weighted towards the pupil vessels. All right, I'm going super quick, but you'll have these slides available. And I already talked this almost to death as far as the Merrick paper, ways of potentially improving your statistical power by better normalization, better selection of the individual subjects, understanding their differences. And I really do think a key problem is the normalization in the spatial maps in the brain, and also doing multivariate analysis and not just using resting state to pull out the data. So I don't know. This is an important one. I just want to mention maybe one of the last ones, and that is clinical, practical clinical implementation. So, and this is something that people don't really think about. When clinicians use MRI, it's really simple. They have a couple of knobs to adjust white matter or T1, T2, contrast, proton density, and out comes an image. And they can play with the window and shading level, window and contrast level. And they see things like tumors or lesions or places where people had stroke or wherever. And it's really simple. With PET scanning, you also just have accumulation of radioactivity in the brain. Boom, it's there. There's nothing you can do about that. But with fMRI, there are hundreds of different ways of making fMRI maps from your time series data that not only do we not all fully agree on, but might actually need to be different depending on what you're looking for. ways of doing, pre-processing and regression choice and thresholding that are not standardized yet. Maybe with machine learning, it might become more standardized, but that's part of the reason, that's part of the problem. The challenge of implementing fMRI clinically is that you can't just have an fMRI map kind of come on the screen and have a clinician just say, oh yeah, this is the fMRI map, there's the thing. Well, one, there is no thing. There is no biomarker that will help identify. It's usually much more subtle than that. Pre-surgical mapping has been used this way, but it's a lot of work. It's a lot of work. But one clinical application of fMRI that might be very useful, among others, is sort of like identifying network areas that might help guide neuromodulation. I think this might be a very low-hanging fruit for fMRI. All right, just about done. We have individual differences. I talked about that. Layer fMRI, it's very challenging. But one big challenge with layer fMRI, it's not only the fact that it's hard to do layer fMRI. That's already given that it's extremely challenging. But to me, the biggest challenge, and something that I'm pushing my own group to try to figure out, is you have this whole brain of data of not only of 3 dimensions. It's sort of like 4 dimensions. It's 3 dimensions. And then you're looking at layers within the layers. It's hard enough with data that has 5 millimeter voxel size to do a massive correlation map on that. You usually divide it into parcels, and that's it. But let's say you're looking at each voxel, and then you're also trying to look at correlations within each layer. How do you cast a net in processing that captures all the information that might contain of different layers communicating with other layers elsewhere in the brain without losing anything? That problem is not solved at all. And it's a computational problem, but it's also a data modeling problem that I don't think we've solved. So the idea here is that if you make a matrix that's super high resolution, you get something like this. But then you can actually take each little network and make a layer dependent matrix of what layers in that network speak to other layers. And that's just parsing out the fractions of the brain with a lot of analysis to look at that relationship. But can we do this all in one big swath of processing to gather all the correlation of resting state or activation signal across layers and across time as it varies over time? Like I said, computationally really hard, and just hard in terms of what features to look for. All right, I think I'm just about done. This is about it. That's subcortical activity, and which is very important. Lymphatic system, and I don't know why in the world I had this. This is a problem. Shimming, practical problem, solvable. Vendors haven't quite done it yet. So, okay, those are the controversies, mysteries, and those are the challenges. And yeah, some are very, very simple. One thing I didn't really mention at the very end is that a lot of the practical challenges in terms of fMRI pulse sequences and things like shimming that are very important to fMRI can be solved if vendors pay a little bit more attention to things like this. But for vendors, for the sequences that they use clinically, the shimming, or how homogeneous the magnetic field is good enough. So they don't really put much more energy into it. So even though fMRI has benefited substantially by vendors improving pulse sequences, having multi-shot, multi-bandwidth, multi-band sort of imaging, parallel imaging, there's things like this that are solvable, either with more shim coils or better algorithms that haven't quite been solved yet because of the fact that it's sort of a vendor level solution that needs to be done. But that's one of the challenges. So in general, if you're a methodologist, you can look at any of those to help guide your research projects, even the mysteries and to some degree the controversies, because all of these things really are a little bit more open-ended in terms of very much open-ended, especially the challenges in terms of what remains to be done. So with that, just finishing up with an old AI-generated various fun with AI several years ago. All right, well, thank you. Thanks.